Thank you for attending today's Lunch and Learn seminar. My name is Tracy McGinney. I work in Shared Resource Management. And first, I just wanted to highlight that this seminar series is supported by Vivian Palladoro, a retired URMC clinical pathology administrator. She's the one who kind of provides the funds to allow us to give you guys all lunch and learn about the different resources available at CART and Wilmot Shared Resources, some upcoming events. In December, we don't have a regular Lunch and Learn seminar. We are hosting a community event for those interested in the shared resources and what they do. It's called the Shared Resources 101. We are soliciting speakers, so if you're interested in presenting your work to a lay audience, the focus would be on the shared resources and how they support your work. But if you're interested in presenting, you can email me for more information. That event is on Friday, December 12th. And then as a final event that Dr. Eichelman will be talking about is there's a spatial workshop. It's part 3 of their series. It's this Friday. And yeah, that's kind of all we have. If you have any questions about the shared resources, you can send me an email. We have our annual shared resource calendar here at the front, so feel free to take one before you leave. And now I'm going to turn it over to Dr. Eichelman, who is a bioinformatics analyst in the GRC, who will talk to you about some omics platforms. Yeah.
All right. Hi, everyone. My name is Hannah. I, like Tracy said, am a bioinformatics analyst with the Genomics Research Center. And today I'm going to tell you about some testing that we've been doing and expanding the offerings that we have for the community. And I first want to highlight that I'm presenting this on behalf of a large group of folks across the lab and other folks on the informatics team that have put a lot of time and effort into all of the stuff that I'm going to tell you about today. And so just want to make sure that I highlight that. We are part of the Center for Advanced Research Technologies, or CART, if you have not heard about us before. And yeah, a really awesome group of folks across both the wet lab and the informatics side that we're all here to help support the research and the exciting work that you do. So today I'll tell you about 3 kind of main points. The first will be going over an evaluation of 5 single-cell RNA-seq platforms that we've tested using patient-derived AML samples. And the second part of the talk will be focused on the genomic shared resource support for a new NGS-based proteomics platform called Olink. And then in the last part, as Tracy already kind of teased, I'll go through a little bit more detail about our upcoming spatial transcriptomics workshop that's happening this Friday. Okay, so we'll start off with the single-cell AML study. So just a really brief introduction to single-cell sequencing. So we talk about bulk RNA-seq, kind of the most popular version of this type of data analysis. Almost like, think of it like a smoothie. You're homogenizing your tissue, which means that you get homogenized expression out of the end of it. So we're not able to really distinguish cell types or what those individual cell types are doing in the context of their expression profiles. And single-cell RNA sequencing gets you a bit more like the fruit that goes into your smoothie, where you're able to take individual cells and group them based on their similarities and type them essentially to be able to understand the differences between those groups. And so you can get really granular with this. For example, like T cells or CD8 T cells, or even more granular like exhausted CD8 T cells. So a little bit more nuanced compared to our bulk RNA-seq smoothie. And finally, where we'll end today with spatial transcriptomics gets us towards the spatial architecture of our gene expression. And so more like the fruit tart and where all of our delicious fruits are arranged in context of space. And so single cell really gets us kind of individual cells, like I mentioned, and the profile of individual cells in terms of their gene expression. And so you're able to get at the heterogeneity and subpopulation expression variability of your samples. whereas that heterogeneity is really masked in the bulk RNA-seq analysis. And so the GRC has evaluated several options for single-cell RNA-seq platforms, including from these companies that are listed here, 10x Genomics, Parse, and Illumina. And so today I'll talk about some kind of head-to-head comparisons that we've been doing against kits from these companies and the work that's come out of that. So from 10x, we've evaluated their GEMX kit as well as on-chip multiplexing, which I'll refer to as OCM for figures in the rest of this section of the talk, and then also their Flex kit. Parse, we've evaluated the EverCode WT version 3, which I'll just be calling Parse from now on. And Illumina, which was previously Fluent, I'll mention here. We tested the Illumina single-cell T10 kit, what they now call the Illumina single-cell T10 kit, or it'll be abbreviated ILMN for the rest of this part of the talk. And so the goal here is really to evaluate these various single-cell platforms to be able to provide informed recommendations to you all, to the community, about these kits and the usefulness for your research. And so a quick overview, kind of head-to-head comparisons again of these 5 platforms. Here's the 5 that I just went over. So 10x, GEMX, OCM, Illumina, Parse, and then the 10x Flex. So there's some variation in the type of technology that they use. So 10x all uses this droplet encapsulation method. And so it's using microfluidics to capture just one cell and one barcoded bead together in a small oil droplet. Illumina uses PIP formation. PIPs are particle templated instant partitions. And so it's using these template particles or hydrogel beads that have barcoded oligonucleotides attached to them. And so when you vortex the cells in the oil, you get these template particles that partition the cells and barcode your template into separate emulsions. And then Parse is using cell compartments and combinatorial indexing in contrast. There's some differences in whether or not you can use fixed or unfixed cells in your input. So the first 3 listed here, you cannot ' fixed cells, but Parse and 10x Flex you can. There's also some differences in the type of profiling that you can do. So the first 3 listed here use 3' and 5' or just 3' expression. For Parse, you actually get the full transcript, and then 10x Flex is the only technology here represented here that's probe-based. And there's also some differences in the capacity, the number of cells that you can input, ranging from 5,000 with OCM up to 10 million for Flex. And just a quick note that the 10x on-chip multiplexing protocol requires a minimum of 4 samples. All right, so here's an overview of the workflow that we've used for this testing. So we're starting with patient-derived AML apheresis samples, 2 of them that I'll be really focusing on for the results I'll show you today. So we are using either fresh captures or direct captures, or fresh cells with fixation for the kits that can take the fixed cells. For all of these methods, we're targeting 5,000 cells. Here we're just showing that we achieved approximately 5,000 cells, or at least for all of these methods. And then we go on to sequencing. And so the vendor recommendation for all of these methods is about 20,000 reads per cell. But here we're showing, in order to compare kind of head to head, what is the difference in the data analysis between these kind of vendor recommendations and the increased sequencing that we usually perform at the GRC. We've bioinformatically downsampled the increased depth of sequencing that we did down to this 20,000 reads per cell recommended depth. And so I'll show you some comparisons head to head between this downsampled data, which is downsampled to the kind of standard recommendations from these companies versus the increased sequencing depth. Okay, so the first look at some of the data that we've generated. Here we're looking at some data quality metrics across the different platforms. So each of the platforms represented here On the x-axis, all of the salmon colors are the recommended depth, or that 20,000 reads per cell, and then in teal is the increased sequencing depth results. So we're looking at the total number of genes detected on the left. In the middle is the percent mitochondrial reads, which is often an indication of kind of compromised cell integrity. And then on the far right, looking at the percent ribosomal reads, again, kind of a measure of cell integrity or RNA complexity. And so what we're seeing here is that for the number of genes detected, the increased sequencing depth for GEM-X and OCM resulted in more genes that were detected. And for the percent mitochondrial and percent ribosomal reads, there really wasn't a significant effect of the depth on these two metrics, these two quality metrics. So next we're integrated our samples and are visualizing the data that we have in a UMAP. And so here we're looking at our two AML samples integrated, and so data for both of the samples is represented here. And when we color the UMAP by the sample ID, you can see here that there's pretty large separation in UMAP space based on the sample identity. And also when we visualize, instead just based on sample, but based on prep type, we can see that there's also some differences that emerge based on the prep type. So here we're integrating not just across samples, but across all the prep types, excuse me, to be able to compare and contrast them in this UMAP space. And so visualizing this a slightly different way as a stacked bar plot, here we're looking at those same clusters, but just numbered on the x-axis, and then the proportion of each of the different kits is represented on the y. So here what we're really seeing is that a few of these clusters, 7, 11, and 16 in particular, are primarily derived from the Illumina kit. Whereas the other clusters are pretty well distributed, where each of the kits or the prep types is represented across those other clusters, which is more what we would hope to see if the kits were all kind of performing similarly. Another visualization of this in the UMAP space, here now we're looking at the number of genes detected and the color. So higher number of genes detected is a darker blue-purple color. And here we're seeing that cells that are in these Illumina-dominated clusters had some of the fewest genes that were detected across all of this integrated data. And so this is, we're not finished up with this testing yet, but what we have learned so far, really overall the platforms are performing pretty similarly with some exceptions for the Illumina clusters like I just showed. But all of them are viable options for single-cell RNA-seq studies, and the platform that you select is really based on factors, including those outside of standard metrics, for example, budget and ease of use and experimental design, study goals. And so we're at the GRC, we're always here to talk to you about these considerations as you plan out your single-cell RNA-seq experiments and think about which kit you might want to use. There's still, I think, a lot more that we can learn from these data. And so some goals for us moving forward with this testing are going to be to perform and compare some cell typing across these platforms to get a sense of any particular cell types that are picked up in one particular method versus another. We'll also work to characterize the genes that are gained from the increased sequencing depth versus the downsampled vendor-recommended sequencing depth. We'll also investigate this Illumina analysis a little bit further, but their workflow is slightly different than the others, and so we want to work a little bit more to remove these low feature cells and kind of reanalyze without those low feature cells included. Also, there's a lot more information to gain from the parse full transcript coverage, including looking at things like isoform expression, alternative splicing, that kind of thing. And then lastly, just as kind of a PSA, we're also looking into long-read sequencing applications for single cell, and something that we'll be thinking a lot about moving forward. Okay, on to part 2 of the talk. So focusing on the NGS-based proteomics platform called Olink. So Olink is a new kind of method that we've been testing and will be offering as well to the community Just up top, I'll say that Olink and some folks from that group are coming in the new year in early 2026, and they'll be presenting even more on this kind of data and how it can help your research. And so keep an eye out for that announcement. But yeah, that'll be early 2026 that they come on campus. So Olink is a complementary approach to other proteomics methods like liquid chromatography, mass spectrometry. It's interesting because it's an NGS-based proteomics panel. It has really high throughput and broad coverage, so it's a great exploratory method, and I think what makes it complementary to other methods like LC-MS. The panel that we've tested and that I'll be telling you about today is called the Olink Reveal panel, and so this panel consists of 1,034 proteins. You can get up to 86 samples per run, and it only requires 4 microliters of sample. So really, they advertise this as a really high performance and broad coverage panel. It does focus a little bit specifically on inflammation processes, and they have a lot of other interesting details about kind of how they selected these proteins on their website as well. So Olink uses this proximity extension assay, which involves an immunoreaction where these 2 matched antibodies are labeled with unique DNA oligonucleotides, and those 2 antibodies are required to both bind to the target protein, represented here by this blue dot. And so when these are in close proximity, the DNA oligonucleotides hybridize, and then they're extended. So you get a DNA barcode that's unique to each of the individual target proteins. So you can then amplify that DNA and sequence it on a standard NGS platform. So this proximity extension assay, or PEA, is really highly specific and has high sensitivity. And so they verified that there's really low likelihood of kind of cross-reactivity or off-target binding using this assay. It's also very scalable, so you can scale across kind of multiple plates, and then you get the same performance that's achieved when the assays are multiplexed. They also have a really wide variety of internal and external controls. They're all listed here. I won't go into each one in depth, but all of these internal and external controls lead to their having a very specific normalization and quality control step in their pipeline. So there's really high fidelity, I suppose, with using these internal and external controls. So once your data, Olink data comes off the sequencer, you get raw counts and then normalized protein expression values, which we'll talk a little bit more about on the next slide. And that's kind of the standard deliverable from Olink, but the GRC and the informatics team will be available to help with kind of all of these downstream analyses that we'll go through, including some QC, differential expression, pathway analysis, and other custom analyses if you need. So these normalized protein expression values are also called NPX values. They're relative to the controls, so all those internal and external controls that I mentioned. So you don't get absolute protein values out of this assay. And so you also are really only able to compare kind of within the one run that you do within the one plate, because they're the relative values. But they're on a log2 scale, so an NPX of 0 essentially means that you have 1 times the control, and an NPX of 1 means you have 2 times the control, etc. Okay, so part of the GRC's downstream analysis pipeline includes quality control. So we have multiple steps in this analysis pipeline that can help identify assays. Olink calls the proteins assays, so that's what I mean here. So assays are samples that don't meet the Olink quality metrics. So a couple of those would be this interquartile range plot. This is all based on synthetic Olink data that I'm showing you here. But this interquartile range quality control step is identifying outliers or samples that fail QC metrics. And so you see here there's red points are indicating a QC warning. And so those could reflect a potential technical issue in the library prep or in the sequencing. And there's actually only one outlier here up at the very top that was identified, which could be due to sample quality. So a way to help you kind of dig into samples that aren't passing the QC metrics. And another way to do that is to visualize the principal component analysis. And so these are, here we're looking at the PCA colored by the two treatments. In teal is the treated samples, and in gold is the untreated. And so it helps visualize factors that drive the variance, but also identify outliers and provide some insight into how the samples are clustering and whether they're clustering like you would expect based on your treatments. Okay, we can also get differential expression and pathway enrichment from these results. So the differential expression here, we're looking at a volcano plot, an individual kind of significant, we're thresholding this based on significance. So red points are significantly differentially expressed. Blue points are non-significant here. This is based on a Wilcoxon test with multiple test correction. And we can also look at pathway enrichment. And so this is also performed based on the differential expression results that we're showing here and using a gene set enrichment analysis, essentially. It's important to contextualize these kind of pathway enrichment analysis results with a note that the Olink panels are all designed around kind of specific biological themes. So important to consider the pathways that might not be represented here based on the proteins that they do include in the panel. Okay, so we had a cool opportunity to compare. We have an example lymphoma data set where we're able to compare these Olink reveal panel results with the results from another Luminex assay. So here we're looking at the 16 samples that we included. 9 were lymphoma samples and 7 were control. Here we're just representing the plate where you have your samples here on the left, and then the Olink controls on the right, which includes your sample controls, plate controls, and negative controls. Also just representing here that you would have a lot of extra space to kind of fill up with more samples if you wanted to. Again, a lot of samples can be included in this assay, which is great. So these 16 samples were, like I said, previously run through a Luminex panel with a flow core. And so this allowed us the opportunity to test kind of the consistency across this Olink reveal panel and then the Luminex panel. Not all of the same proteins are represented across those two panels, so we're going to be focusing on the 5 that were shared across the Luminex and the Olink data. So here we're looking at expression in those lymphoma samples versus control samples. Positive expression here, increased expression in the lymphoma samples is to the right of 0, and then decreased expression in the lymphoma samples compared to the control is on the left. So we see 4 of the proteins that are upregulated essentially, or increased expression in the lymphoma samples, and then 1 which is decreased. And so IL-6 is the only significantly differentially expressed protein after the multiple test correction that we do. But digging a little bit more into comparing the directionality of those results across the 2 panels, we'll go through all 5 of those proteins, but just to orient you, we're looking at the results from the Luminex assay on the top and the Olink reveal assay on the bottom. Control is still in gold on the left, and then lymphoma in teal on the right. So first, for GFAP, the results were consistent in terms of directionality, where it showed decreased values, expression in the lymphoma compared to the control. In IL-10, similarly, the directionality was consistent, where we saw an increase in the lymphoma samples compared to the control. For TNF-alpha was fairly consistent in terms of there wasn't much change across the treatments here for either assay. And same for IL-4. IL-6 was a little bit different, where we saw kind of the expected increase using the Olink reveal panel in lymphoma compared to control, but there really wasn't much change across the treatments using the Luminex panel. So in summary, most of the assays of interest, the ones that are shared across these 2 panels that we can compare, reveal this consistency and the directionality of expression across the Olink reveal and Luminex. And Olink reveal shows us the kind of expected pattern in IL-6 of increased expression in the lymphoma treatment. And although these cross-platform correlations aren't exactly perfect. They're quite good, and I think provides additional evidence that Olink is a complementary approach for other proteomics methods that are available. And so another kind of quick summary of some other aspects of these panels, this Olink reveal panel in comparison to LC-MS and mass spectrometry. So the sample type is a pretty important difference where Olink takes these intact proteins. LC-MS requires the digested proteins. For Olink, you just have to dilute these intact proteins, but LC-MS is a bit more involved with digestion, reduction, etc. There's differences in processing time, much shorter for Olink compared to LC-MS. And again, for Olink, you're limited for this reveal panel to the 1,034 proteins that are included in that panel, whereas it's much more dependent on the upstream workflow and the sample type for LC-MS. For Olink, because they have all of these internal and external controls, they've published and shown that you really need just one sample replicate to get an accurate representation of the proteins that are represented, whereas for LC-MS, there is at least 3 that are recommended. And then for data analysis, Olink does have a pretty handy software to get those NPX values, plus we're here to help you with kind of all downstream analysis that you might want to do. Whereas LC-MS requires some more extensive training compared to Olink. And so, yeah, this Olink NGS approach is really helpful for data exploration, and the proteomics core with LC-MS methods can really help follow up and validate these findings that you get out of the Olink panel. Okay, so I will wrap up with a preview of the upcoming spatial transcriptomics workshop. So this Friday from 10 a.m. to 2 p.m., we'll be going through some 10x Visium and Xenium analyses. We're digging into kind of cell typing, spatial pseudotime, cell-cell interactions, and some other really exciting techniques for analyzing your spatial data. This is the third seminar or third workshop in our spatial seminar series. And so we also have materials available from the first 2 workshops that we're happy to send out. And so the first part of the first workshop, we really went over kind of the different methods that we have available at the GRC, both sequencing based with Visium and Geomics, as well as the imaging based method that we have, which is 10x Xenium. And so we also went through kind of the standard analysis that you can do with these methods in the second part, our second spatial workshop, where we went over pre-processing the data in ScanPy and SquidPy. So we went from kind of how do you read in the data, normalize the data, and perform dimensionality reduction and clustering, generate your UMAP and visualize in space, and then get to differential expression across your treatments. And so on Friday, we'll be going through kind of more downstream analyses with this package called ST-Learn. And ST-Learn is designed to comprehensively analyze spatial transcriptomics data. It leverages spatial distance, your tissue morphology, as well as gene expression from your data. And it allows for more accurate modeling of underlying tissue biology, and addresses these really cool questions in terms of cell trajectory reconstruction and cell-cell interactions and intact tissue, among others. So you can scan the QR code here to learn more and also to register, but hopefully we'll see some of you there on Friday. So that is all that I have. I've included emails for the bioinformatics team and also for myself here. Please don't hesitate to get in touch if you have any questions about anything I talked about today, and also a QR code to learn more about the GRC in general. I'm happy to take any questions. Yeah.
Earlier you showed kind of a plot that comparing between 2 groups in the Olink, comparing to the Luminex. I was just curious, because it seems like your Luminex is like generally is more skewed compared to the lymphoma groups compared to the Olink. Is that something that's from the method itself, the normalization, or the QC, or the mix of three?
Yeah, do you mean that the values, there's more variance in the data, or sorry, what do you mean by?
So IL-10, IL-6,
Mhm.
it seems like they have like more of like a, you can see it's more like a grouping at the bottom.
Mhm.
Whereas like if you look at the Olink, it looks like there's a little bit more scatter that would allow more homogeneity, that would allow for better normality properties. Is it something that's just from the method itself, or is that like a normalization? An aspect from
Yeah.
normalization?
I'm admittedly not a Luminex expert, but I would imagine it's something inherent to the method, but I wouldn't be able to say that for sure. Yeah, the normalization is really all the kind of internal and external controls, and that normalization I went through is specific to the Olink panel. And so in terms of how these are also just for context, like an absolute value. So this is in picograms per milliliter, whereas we're looking again at the normalized protein expression values here. So it's a different scale too. But yeah, I couldn't say for sure, to be honest, what is causing these kind of points to be bunched up down here at the bottom. Yeah, but Jeff might have.
I'll just jump in. So we collaborate with a lab, who ran these, and they cherry picked these samples for us to validate the Olink on. So I'm not exactly sure the decisions on which samples were chosen, but they're Luminex based on the Luminex data. So this isn't a full representation, and certainly we're not here to sort of say Luminex is bad and it's good.
Yeah.
I think they each have their place. You know, Luminex is absolute. There's controls on to get these, picograms per microliter concentration versus Olink, very difficult to measure, right? So, yeah, I don't think we're equipped to go to sort of go into depth on Luminex data calculations or that, but they're complementary approaches, and part of this analysis limited based on the samples that we were given to sort of comparison.
So when you run this, do they have like different comparison here?
There's standards for the next sample.
Mhm.
So no, that's separate. Here, one's equal next. Yeah, my understanding, you have to understand that you might have translate the signal you get from the Luminex assay to absolute values. Whereas Olink is a very different measure, and it's all relative. All the same, etc.
But on the Olink again, I think you're saying you couldn't compare from plate to plate, just like if you had more than
Unless you
100 samples.
Unless you had samples across those plates, if you kept the same samples across them, then you would be able to compare. Yeah, exactly.
So I think they definitely do have it. They do have the plate controls that allow you to randomize your samples across a larger study like that. So it is designed with that in mind. So the reveal assay that we're offering is specific to that 9612 plate format. Olink has other platforms that require automation that we don't currently support, that require a lot of capital investment, that can run many more assays and more samples, but you can run a 500 sample study across 5 or 6 plates and be able to do this type of analysis if they're delivery controls or something.
Yeah.
I had a question about the single cell part at the beginning. I wonder if you could just say a little bit about like the cost per cell between the different platforms, and also with regard to the Illumina where you had those additional clusters. How can you be sure that those are kind of low quality that you don't want versus cells that are being missed by all the other platforms?
Yeah, absolutely. I think I'll start with your second point, because I think that gets at the cell typing future direction as well. So you're right, there's absolutely cell types that could be valid cell types that have fewer genes that are detected, and so we wouldn't want to remove those. But part of the kind of comparison there will be, typing the cells and seeing if it's actually a valid cell type that we're catching here with Lumina, or whether it is true kind of low information cells that we would want to remove earlier in the processing. Yeah.
I don't know, cost. That's the thing I told you to take out. Do you think going back to your chart that
Yeah.
summarizes everything?
there's so many factors that kind of like influence cost of the cell, right? So like even when you look at those fixation methods, those cell numbers are what you need to do the fixation, not necessarily what you're going to capture. And also like we did 5,000 in this test, but you know, you could do up to 20,000. The read depth that you do, all of these things impact cost. But in general, you can say fixation methods and multiplexing methods are usually more affordable because you can fix and multiplex and put together many samples into one capture. Your GEMX is going to be your most expensive per cell just because you're taking one sample, unless you use hashing methods, which is a whole nother conversation that influences cost. One sample per capture, basically. So ultimately, there's all these forks in cost discussions that kind of lead us one way or the other. But you can think multiplexing is usually more affordable than multiplex, and then GEMX being the most. But yeah, we give you cost when we talk about your experiments. Try to be transparent as much as we can.
Great. Thanks all.
Good job.
Thank you.
Good strategy.